Today, me and Bethan are going to talk about biochemistry. The first topic I'm going to talk about is the three major biomolecules. There is carbohydrates, lipids, and proteins. Carbohydrates are made up of carbon, hydrogen, and oxygen using the formula CH2O. The biomolecule is made up of monomers and polymers. What are the difference? Monomers are singular building blocks, which larger molecules are made from. Once joined together, they become polymers. Polymers are always three or more. They can go on for thousands. These monomers are like building blocks. They are replicated repeatedly to make the polymers. The monomers found in carbohydrates are monosaccharides, which are simple sugars. The most common simple sugars are glucose, fructose, and galactose. As you can see by the diagram, the monomers use the same carbon, hydrogen, and oxygen. However, they are slightly different to make them unique. These can be joined together to make disaccharides. These monomers are joined together using a glycosidic bond. Once the bond is together, this is when a monosaccharide becomes a disaccharide. As I've said, disaccharides are two monomers joined together. As you can see, we've got glucose and we've got fructose joined together by the glycosidic bond to make sucrose. There is also other combinations such as glucose and galactose to make lactose, which is the dairy, and there's glucose and glucose, which makes maltose, which can find in wheat, barley and fruits. When you join three or more monomers together, it becomes a polysaccharide. This is where you join it by a glycosidic bond using a condensation reaction. Using a condensation reaction makes this insoluble in water. This can then be broken down into monosaccharides by hydrolysis. This is where you add water molecules for the ones that have been lost. The types of polysaccharides are glycogen, starch, and cellulose. So now we're going to talk about lipids. There is fats, waxes, and oils. Lipids are hydrophobic and lipophilic. This means they hate water, but they love fat. This makes them insoluble in water. Lipids are three fatty acids attached to the glycerol molecule. This makes them a triglyceride. Triglycerides are usually fats and oils. Removal of one fatty acid and replaced with a phosphate group makes a phospholipid. Phospholipids are very important in cells. Like discussed before, lipids play a big role in cells. They are used to build up the phospholipid bilayer, which is the cell membrane. As you can see by the diagram, hydrophilic head and a hydrophobic tail. This is how they arrange the cells in the cell. This makes up the cell membrane and controls the molecules what go in and out of the cell. Another function is long-term energy storage. This is used once all the carbohydrates are burned off. The last biomolecule is proteins. Proteins are made up of amino acids. There is a variety of 20. These amino acids are formed to make dipeptides and polypeptides. Each peptide chain has a different R group, which makes them unique. Dipeptides are two monomers. Polypeptides are more than four. As you can see by this diagram, this is a polypeptide as it has more than 4 amino acids. Proteins are made up with amino acids and carboxyl groups to make the peptide bond. As you can see by the diagram, we've got one amino acid and another amino acid separately. These are the monomers. This is then joined by a peptide bond using the condensation reaction. This is a removal of water. As you can see, these are the R groups, and there are different R groups per peptide bond. This makes them different and unique. This is then classed as a dipeptide as it has two monomers joined together. If you was to add more, this would then make a polypeptide. When it comes to proteins, there are 3 different structures of polypeptide chains. You've got primary, secondary and tertiary structure. As you can see, this is the primary structure of amino acids. Then you get onto secondary structure, which is an up-down wave, or it can be also a zigzag. This is called B-pleated sheets, or this one is called alpha helix. These are bar groups which have a slight positive charge and a slight negative charge, which draws them together to make these unique shapes. A tertiary structure is a complex 3D shape formed when A helix and a B pleated sheet folds around themselves. This is when the R groups have an attraction between one another, which causes these 3D shapes to appear. Functions of proteins are fibrous. This is the structural function. These provide strength and protection to cells and tissues. Also, globular proteins are used for metabolic function, such as hemoglobin. Now on to the next bit. The next topic we're going to talk about is enzymes. Enzymes increase the rate of chemical reactions. They have ideal conditions to be efficient. They break down lipids, starch, and proteins. Now Bethan's going to talk about this topic in more detail.
Thousands of different chemical reactions must happen in our bodies in order for it to function. Enzymes are made up of thousands of amino acids linked together by peptides or polypeptides. These are all linked together in specific ways to form different enzymes. Enzymes are biological catalysts. They're highly specific. They are 3D in structure and they must bind to a specific substrate for a reaction to happen. Activation energy means the minimum amount of energy that is needed for a reaction to take place. So if the activation energy is high, the reaction will be slow, and the more energy that will be needed, the slower it will happen. If the activation energy is low, then the reaction will be quicker, and the least amount of energy needed for a quicker reaction to happen. Enzymes or biocatalysts can either increase or decrease the rate of a chemical reaction. So, in 1984, Emil Fischer, a German chemist, proposed the term lock and key. He stated that enzymes must fit each other like a lock and key in order to have any chemical effect on each other. Enzymes have a certain shaped area called an active site. This is where bonding happens. The substrate, which is the chemical that is interested in the enzyme, is what connects or bonds to the active site. If you think of a lock and key, like in the image, that's how they fit. In 1958, Daniel Koshland, an American biochemist, proposed a variation to the lock and key model. He suggested that bonds could be more flexible, and he named this the induced fit model. So as we know, this is similar to the lock and key model. Koshland believed that the bond between active sites and substrates was not rigid and that the active site could change shape to fit the substrate perfectly. Inhibitors play an important part of the chemical reaction between an active site and a substrate. They reduce the rate of a reaction and they make sure the reaction doesn't happen too quick. Two other inhibitors to talk about are non-competitive inhibitors and competitive inhibitors. Non-competitive don't bind to the active site, they bond to a different area, and once the molecule has changed the shape of an active site, the enzyme becomes conformational, meaning it can't be used again. Competitive inhibitors, they're similar to substrates. They will temporarily bind to an active site, and this happens when two substrates are challenging for the same active site. These enzymes can be used again. Something else to talk about is cofactors and coenzymes. These may bond to the active site or substrate to help the enzymes carry out its job by breaking up or building up substrates into products. They help the active site become the perfect fit for the substrate. Cofactors could be metal ions and coenzymes could be something like vitamins. Enzymes do not like the cold. In fact, they are exceptionally sensitive to thermal changes. The measurement of temperature is done by how quickly molecules move, leading to collisions. This is also known as kinetic energy. As the temperature rises, the rate of enzyme activity will rise too. The optimum temperature for optimum enzyme energy is between 37 and 40 in humans. If the temperature continues to rise and then suddenly slows down and decreases, the enzyme's active site will become distorted and the enzyme will become denatured. If an enzyme becomes denatured by heat, the bonds break and the enzyme can no longer be used. So just like enzymes needing an optimal temperature, they also need an optimal pH level. The pH level for most humans is between 4 and 8. A pH level can also affect the shape of an enzyme's active site. Extreme changes in these levels can cause an enzyme to lose their 3D structure. If the enzyme level continues to increase and then decrease again, enzymes will become denatured, and as we know, this means they cannot be used again. Enzymes work at their optimal when there is plenty of substrate available. They use the least energy, therefore there will be increased reaction. So lots of substrates around make it easier for enzymes to fill their active sites. The more substrate available, the more enzyme activity that will happen. Therefore, the less substrates make it harder for enzymes to fill their active site and create products. They will use more energy as they have to move around more to find a substrate to fill the site. If the concentration levels increase or continue to increase, the reaction rate will remain the same.